Good afternoon, guys, and today we will discuss about some of the most important steps of glycolysis. And in the previous lecture, we have discussed about the difference between glucokinase and hexokinase enzymes, how they work, what are their Michaelis constant and their Vmax, etc. You can watch through that video. And today we will discuss about the most important steps in glycolysis and some revision. So, first of all, when glucose enter into the cell through the activity of transporters, it is trapped inside the cell. It must be trapped because these transporters are bidirectional transport mechanisms. So, if these are not trapped, they can move out. It means that glucose can move out of the cell if this is not trapped. The glucose is trapped in the form of glucose 6 phosphate. We are attaching a phosphate group to that glucose, and in this way the glucose is trapped. And we are adding phosphate group. It means that we need phosphate. The most important source for the phosphate is adenosine triphosphate, ATP. And when ATP yield one phosphate and gives to glucose, it is left with ADP. It is adenosine diphosphate. And this step is very critical because it's irreversible step of glycolysis, and it's under the influence of these hexokinases and glucokinases. I have made a detailed video on these two enzymes in the previous lecture. Okay, after that the glucose 6 phosphate is converted into fructose 6 phosphate. It's a reversible step, and it means that it can go in either ways. And this step is by the activity of phosphoglucose isomerase. It's an isomerase enzyme because these two glucose 6 phosphate and fructose 6 phosphate are hexoses, just interconversion, because these are isomers, so the isomerase enzyme is used. The most critical and important irreversible step in glycolysis is the conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Okay, it's very important, and it's a irreversible step undergone by the enzyme known as phosphofructokinase 1. PFK1 has many pockets. For example, it has many regulations, allosteric regulations. It has a pocket for ATP, it has a pocket for citrate, it has a pocket for AMP. ATP and citrate levels indicate that the cell is high in energy. So, do we need glycolysis in that case? No, we don't, because already the cell has ATP. AMP represents that the ATP is broken down and the cell has not sufficient ATP. So AMP is a positive regulator for glycolysis. The most important regulator for glycolysis is the fructose 2,6 bisphosphate. It's a substance or molecule. It is produced from fructose 6 phosphate. For example, if there are millions of fructose 6 phosphate, hundreds of them are converted to fructose 2,6 bisphosphate, and the other millions are converted into fructose 1,6 bisphosphate. But this is a very powerful regulator because it regulates the PFK1 to convert the remaining fructose 6 phosphate to F16BP. This enzyme is regulated by a hermaphrodite enzyme. It means that it has two activities, two domains. One is the kinase domain and other is the phosphatase domain. Kinase activity is to phosphorylate something. Kinase activity converts the fructose 6 phosphate to fructose 2,6 bisphosphate, and phosphatase activity is to dephosphorylate something. Phosphatase converts fructose 2,6 bisphosphate back to fructose 6 phosphate. Okay. Also, one most important thing here to mention is the involvement of the hormones insulin and glucagon. When the blood glucose level is high, the insulin automatically goes up. But when blood glucose level decreases, for example, we have not eaten anything, and in this condition, the insulin level decreases and the glucagon level increases. Glucagon influences this glycolysis in various aspects. One of them is that glucagon binds to a 7-pass receptor and it's bind to G stimulatory, and these all process I have discussed in the previous lecture. I'm just revising that stuff. The G stimulatory influences adenylate cyclase and it converts ATP to CAMP, and it forms the protein kinase A. And what I have told you that kinase enzyme is to phosphorylate something. It means that it's going to phosphorylate this hermaphrodite, this enzyme which has two domains. When this enzyme is under phosphorylated state, its kinase activity stops and its phosphatase activity goes on. It means that it converts fructose to 6-bisphosphate back to fructose 6-phosphate. It means that this was the powerful regulator for glycolysis, and this concentration is decreased and F6P is increased, so glycolysis will not proceed forward. But when the glucose level is high, insulin level is also high, and insulin affects the this enzyme or the glycolysis pathway via the phosphatases. What is the function of phosphatases? Phosphatase function is to dephosphorylate something. It dephosphorylates this enzyme. It removes the phosphate from this enzyme. And when this enzyme is in dephosphorylated state, its kinase activity will work and phosphatase activity will not work, and kinase activity will convert fructose 6-phosphate to fructose 6-bisphosphate, and it was a powerful regulator for glycolysis, and glycolysis will proceed forward. So you can think that how nature and set up this influence of the hormones to the glycolysis pathway moving forward. But the second ATP is yielded here in this step. Okay, the first ATP was invested in the conversion of glucose to G6P. Okay, this is the investment phase, and the second ATP is invested in the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. It's the second most important irreversible step of glycolysis, and it has many regulations. So let's begin our today's topic. It's the conversion of fructose 1,6-bisphosphate to two molecules. It's a 6-carbon molecule and it's converted into two 3-carbon compounds. First is the DHAP, it's dihydroxyacetone phosphate, it's a 3-carbon compound, and the second is the G3P, glyceraldehyde 3-phosphate, it's also a 3-carbon compound. Okay, as you can see here that phosphate is attached to the carbon number 1 and carbon number 6 in the fructose 1,6-bisphosphate. But in case of dihydroxyacetone phosphate, the phosphate is attached to carbon number 1 and it splits. And in case of glyceraldehyde 3-phosphate, the phosphate is attached to the carbon number 3. Okay, but here the most important thing occurs that glycolysis to be processed, only this molecule can function. It means that only the glyceraldehyde 3-phosphate can undergoes the other processes. The dihydroxyacetone phosphate is converted into G3P. It means that the steps which are occurring on this side is also occurring simultaneously on this side. So we can say that from one glucose molecule we have obtained glucose 6-phosphate, we have obtained fructose 6 phosphate, we have obtained fructose 1,6 bisphosphate, and after that we have obtained two molecules of glyceraldehyde 3 phosphate, because dihydroxyacetone phosphate has been converted into G3P, okay, by the enzyme triose phosphate isomerase. These are triose, means these are three carbons. These both have phosphate, and isomerase means that these compounds have structural similarity, okay, so these are isomerases of each other, isomers of each other. So moving on, we have two molecules of glyceraldehyde 3 phosphate. It's very important to remember that here we have two molecules of G3P, because the dihydroxyacetone phosphate has been converted to G3P, okay. After that, up till this we have invested ATPs. We have invested how many ATPs? We have invested one ATP here and the second ATP here. From now on, it's called the harvesting phase or yielding phase of glycolysis. We will now obtain some amount of energy. It's like a business when we invest some amount of money and then we obtain a lot of money through that process. The conversion of G3P to 13BPG. What's 13BPG? It's 1,3-bisphosphoglycerate. It's 1,3-bisphosphoglycerate, okay, or diphosphoglycerate, you can say. It means that phosphate is being added here, and we have two molecules of it, okay. Don't forget that from now on we have two molecules of each substance, okay. We have two G3P, we have two 1,3-bisphosphoglycerate, and in the conversion we will we will harvest the ATP. How it will gonna occur? In this process, NAD positive is converted into NADH. 2NADH, 2NAD positive is converted into 2NADH by the action of enzyme known as glyceraldehyde 3 phosphate dehydrogenase, okay. Just remember glyceraldehyde 3 phosphate and the enzyme is the dehydrogenase, okay. And this enzyme has several pockets. This enzyme has several pockets. One is binding to the inorganic phosphate, okay, and the second is binding to the NAD positive, and the third is binding to the substrate, which is glyceraldehyde 3 phosphate. We have obtained 2NADH, and this 2NADH will gonna yield us ATP. How it will gonna yield us ATP? It will go to the mitochondrial inner membrane, which is the electron transport chain, and in the presence of oxygen and mitochondria and these molecules, it will gonna produce 3 ATP per NADH molecule. This is an indirect step. We will gonna discuss the electron transport chain in detail, but just now believe that these two NADH will go to mitochondria and they will yield ATP there. They will yield 3 ATP molecules per NADH. It means that how many ATPs we are obtaining from this step? It means that 2 NADH, so we are obtaining 6 ATPs. So up till now we have invested 2 ATPs and we have yielded 6 ATPs, okay. And our lecture ends here because you have to revise all of that stuff, and we will continue from 1,3-bisphosphoglycerate onward, but up till now we have investment phase. 2 ATPs are invested and we have obtained 6 ATPs via the electron transport chain. Okay, the same thing is written here that two ATPs are invested. One is the conversion of glucose to glucose 6 phosphate. The second is conversion of fructose 6 phosphate to fructose 1,6 bisphosphate. Yielding phase, conversion of glyceraldehyde to 1,3-bisphosphoglycerate. We have converted NAD positive to NADH, and this NADH will go to electron transport chain and will yield 6 ATPs, 3 ATP 3 ATP per NADH. So this was for today, and in the next lecture we will discuss further processes of glycolysis. And thank you for watching. I will meet you in the next